The baseline rule of biology is uncompromising. All organic matter will rapidly break down and decay when exposed to the elements. In the Italian town of Lanciano, an artifact from the 8th century has remained in the open air for 1,200 years. Kept inside this cathedral, the material is identified as human flesh and blood that has stayed physically intact since the early Middle Ages. While this survival was accepted as a matter of faith for over a millennium, it was finally subjected to a formal forensic investigation in 1971. If this tissue followed the standard laws of nature, it would have crumbled into dust centuries ago, leaving a physical presence that requires a biological explanation. Dr. Odoardo Linoli began his 1971 analysis by treating the relic as a forensic mystery, using the lab equipment of the era to determine if the tissue was authentically preserved. Linoli's method involved testing 3 specific earthly hypotheses that could account for the survival of human tissue over such a long duration. The first hypothesis considered whether the tissue was preserved using ancient mummification techniques. The lab results showed no traces of the mummifying salts or resins that would be required for that type of preservation. The second hypothesis looked for modern chemical intervention, such as the use of formaldehyde. Testing confirmed that no artificial preservatives or chemical agents were present in the samples. By ruling out both ancient mummification and modern chemicals, the study eliminated the 2 primary ways organic matter is intentionally kept from rotting. The third hypothesis examined the environment. Had the tissue been kept in a hermetically sealed or vacuum-packed container? The records and physical inspection confirmed the tissue was fully exposed to the atmosphere and common biological agents for 12 centuries. After failing to find evidence of preservation, the lab shifted to identifying exactly what the surviving material was. The tests positively identified the sample as human myocardium, the specific muscular tissue that makes up the wall of the heart. Further analysis confirmed the accompanying reddish substance was human blood. Finding intact heart muscle and identifiable blood after 1,200 years of open-air exposure contradicts the expected timeline of biological decay. This chart shows the protein proportions of fresh human blood alongside the Lanciano sample. Despite the age of the relic, the proteins are proportioned normally. The sample retains the signature of fresh blood, a state that cannot be maintained naturally over a millennium. Father Spitzer observes that because the samples lack salts, chemical treatments, or airtight seals, there is no biological framework to explain their current state. The evidence suggests a subject that functions outside the known processes of organic breakdown. The 1971 study suggests that while modern forensics can identify the tissue, the data confirms the relic exists in a state that biology cannot replicate or explain.